10.5 Prokaryotic cell division. By the end of this section, you will be able to do the following. Describe the process of binary fission in prokaryotes. Explain how FTSZ and tubulin proteins are examples of homology prokaryotes, such as bacteria, produce daughter cells by binary fission. For unicellular organisms, cell division is the only method to produce new individuals. In both prokaryotic and eukaryotic cells, the outcome of cell reproduction is a pair of daughter cells that are genetically identical to the parent cell. In unicellular organisms, daughter cells are individuals. To achieve the outcome of cloned offspring, certain steps are essential. The genomic DNA must be replicated and then allocated into the daughter cells. The cytoplasmic contents must also be divided to give both new cells the cellular machinery to sustain life. As we've seen with bacterial cells, the genome consists of a single, circular DNA chromosome. Therefore, the process of cell division is simplified. Karyokinesis is unnecessary because there is no true nucleus and thus no need to direct one copy of the multiple chromosomes into each daughter cell. This type of cell division is called binary, prokaryotic, fission. Binary fission. Due to the relative simplicity of the prokaryotes, the cell division process is a less complicated and much more rapid process than cell division in eukaryotes. As a review of the general information on cell division we discussed at the beginning of this chapter, recall that the single, circular DNA chromosome of bacteria occupies a specific location, the nucleoid region, within the cell, figure 10.2. Although the DNA of the nucleoid is associated with proteins that aid in packaging the molecule into a compact size, there are no histone proteins and thus no nucleosomes in prokaryotes. The packing proteins of bacteria are, however, related to the cohesin and condensin proteins involved in the chromosome compaction of eukaryotes. The bacterial chromosome is attached to the plasma membrane at about the midpoint of the cell. The starting point of replication, the origin, is close to the binding site of the chromosome to the plasma membrane, figure 10.15. Replication of the DNA is bidirectional, moving away from the origin on both strands of the loop simultaneously. As the new double strands are formed, each origin point moves away from the cell wall attachment toward the opposite ends of the cell. As the cell elongates, the growing membrane aids in the transport of the chromosomes. After the chromosomes have cleared the midpoint of the elongated cell, cytoplasmic separation begins. The formation of a ring composed of repeating units of a protein called FTSZ, short for filamenting temperature-sensitive mutant Z, directs the partition between the nucleoids. Formation of the FTSZ ring triggers the accumulation of other proteins that work together to recruit new membrane and cell wall materials to the site. A septum is formed between the daughter nucleoids, extending gradually from the periphery toward the center of the cell. When the new cell walls are in place, the daughter cells separate. 10.5 Prokaryotic cell division. 271 Figure 10.15. These images show the steps of binary fission in prokaryotes. Credit. Modification of work by McStruther, Wikimedia Commons. Evolution connection. Meiotic spindle apparatus. The precise timing and formation of the meiotic spindle is critical to the success of eukaryotic cell division. Prokaryotic cells, on the other hand, do not undergo karyokinesis and therefore have no need for a meiotic spindle. However, the FTSZ protein that plays such a vital role in prokaryotic cytokinesis is structurally and functionally very similar to tubulin, the building block of the microtubules which make up the meiotic spindle fibers that are necessary for eukaryotic nuclear division. FTSZ proteins can form filaments, rings, and other three-dimensional structures that resemble the way tubulin forms microtubules. Centrioles, 272 Chapter 10, Cell Reproduction. Access for free at openstax.org. And various cytoskeletal components. In addition, both FTSZ and tubulin employ the same energy source, GTP, guanosine triphosphate, to rapidly assemble and disassemble complex structures. FTSZ and tubulin are considered to be homologous structures derived from common evolutionary origins. In this example, FTSZ is the ancestor protein to tubulin, an evolutionarily derived protein. While both proteins are found in extant organisms, tubulin function has evolved and diversified tremendously since evolving from its FTSZ prokaryotic origin. A survey of meiotic assembly components found in present-day unicellular eukaryotes reveals crucial intermediary steps to the complex membrane-enclosed genomes of multicellular eukaryotes. Table 10.3.